Great. Yes, indeed, we see you. Great. So my pleasure to introduce Dr. Robert Schwartz. He was a professor here in it's really in systems biology and physiology, but also livers, clinical trials, virology, many things he does all the same time. So we'll hear about some of them today, particularly some of the spatial technologies. We've had the pleasure of working with Dr. Schwartz for several years. So thanks for coming and take it away.
Well, again, thank you for the introduction and for the opportunity to speak to you guys today. So I'm going to talk a little about spatial genomic technologies. Some of these slides Chris will be familiar with because I stole at least a few at the beginning from one of our, well, for me, former students, one of his mutual students now. And then I'm going to give you a few vignettes. So basically, I'm just going to give you some background and motivations for the approaches that we've taken and that kind of just discuss spatial transcriptomics and proteomics and then focus on two vignettes. Now, as Chris mentioned, I'm very focused on the liver, and the goal of my lab is to really try to build a bioartificial liver for transplant. And, the thing is, though, when we actually focus on the liver is kind of a very unique organ. So when the parenchymal cell type, the hepatocyte, is injured, basically the way the liver responds is through the proliferation of undamaged hepatocytes. However, when this process occurs too acutely, it results in a process known as acute liver failure. And when it occurs chronically, it results in a process that we know as cirrhosis. And due to the architectural, structural, molecular, cellular changes that occur in chronic liver disease, this causes a lot of morbidity, mortality globally. So basically, in the United States, about 20 million Americans live with some form of liver disease. It's actually higher in Europe, about 30 million Europeans. 3 million Americans have this end-stage process of liver disease known as cirrhosis. About 7 million Europeans have this end process. I should have updated these numbers, but the numbers would be about the same. It's around 10,000 are currently listed, and 9,500 were transplanted last year. And the number just came out they just tallied the numbers for 2023. It's pretty much in line. Now, if you look at this, you're like, oh, that matches up pretty closely. So maybe we're not doing so badly on the transplant side. But the reality is, this number is a little deceiving. So the thing is that there's been a massive expansion in the number of transplants we do in the United States. Historically, it's been somewhere around 6,000 to 6,500. And that number was stable for about 15 to 20 years. And then what changed about maybe 9 years ago, was the opiate epidemic. And that has been really great for my patients because now it's created a huge opportunity for good organs to be made available. Obviously, something pretty horrible for society, but good for my patients. And if you take a big picture step at this, obviously this is not a sustainable source of tissue for organ transplant. Now, despite this, because of this gap and also the fact that there is not a lot of availability of organs, there are a lot of patients that are never listed for transplant. And so 31,000 Americans and 170,000 Europeans die each year from acute and chronic cause liver disease, and worldwide over 1 million people die from cirrhosis. So this is the 14th leading cause of death globally. But actually, if you look at specific causes of liver disease, like Hep B, for example, is the one of the #1 infectious disease killers globally, and one of the most common causes of cancer as well. So, oh, this is going the wrong way. Okay. So obviously, if we could actually build an artificial liver tissue or derive cells in large numbers, you can imagine this could be used for a variety of different uses in both basic science and clinical practice. On the basic science side, let me put the pointer on here. We could use this as tools to study human disease or human physiology, or if we use this in the clinical arena, this could be used to restore or replace lost organ function. And basically, obviously, insights in the basic science arena can feed into the clinical arena and vice versa. And all of this is with the goal to try to improve quality of care for our patients. So with this all said, if we want to go about trying to build a bioartificial liver tissue for transplant or try to do this, really what we're trying to build is the human liver. And it has a very complex architecture, both a variety of different cell types to make up this organ. And if you want to break it down into its most basic components, really, we need to come up with the cells that make up the liver and then also build the microenvironment that those cells live in. And obviously, the different decisions or components that we're thinking about really will ultimately impact kind of the ultimate global phenotype and function of this. But one of the fundamental challenges, though, that we have had is how do we actually go about doing this? We actually don't know much about the architecture of the liver. Obviously we can get H&Es and things like this, but we don't really understand how the cells are well organized within that. And one of the things that is very clear is, and I'm not going to show you some slides of this because maybe I shouldn't have taken granted you guys know this, but in the liver itself, oh, come on, really? Okay, as you can see here within this architecture itself, these hepatocytes that are over here as compared to the hepatocytes here, they're all different. So although the tissue looks like, oh, it's just a bunch of 95% of the cells are hepatocytes, those hepatocytes are actually all quite different from each other. And that difference is kind of dictated based on its organization within the liver tissue itself. Now, what drives what that process is known as zonation. What drives that is unknown or largely not well understood. And it's been partially part of the reason for this is due to the fact that we just haven't had the tools to really dig into this deeply to kind of delineate the molecular drivers of these processes. And maybe just a little side bit just on the liver, just because I think it is interesting. One of the things that makes the liver different from other organs and why, again, understanding the architecture, cellular molecular changes going on within it really could dictate its function, is the liver blood flow is quite different from every other organ in the human body. So most organs, you all learn this, arteries bring blood from the heart, it's oxygenated, it goes into the organ then, goes through the capillaries, it gets deoxygenated, nutrients get taken up, wastes get deposited, it goes to the vein, then brought back to the heart. Okay, that's not what happens in the liver. The liver, only 30% of its blood flow is from an artery. 70% comes from the portal vein, which basically is pulling out all the junk, deoxygenated blood, waste from the colon and the small intestine and actually the whole GI tract. And all of that gets dumped into the liver where then it gets processed. So if you were to think about it, the organization of different proteins, enzymes, detoxification enzymes, all needs to be organized, because if you break something down into metabolite X, then how do you process it now into metabolite Y? One cell can't possibly have everything inside of it to do everything. And so the way the liver is organized is it kind of passes it along. So that, if the blood flow is coming this way, going down here, the cells here will express enzyme X, then the next one will express enzyme Y, and the next one will express enzyme Z. So it kind of passes it along. So this organization is actually critical to normal function of the organ. And again, a lot of this is missed if you, just are looking more globally at it. Okay, so how do you kind of delineate the cellular, molecular, and spatial organization of the tissues? And stole this slide from one of Chris's students. I don't. Have you guys seen this or talked about this already? Or you might have.
Not much. No, so this is good. No, that's perfect.
Okay, good. So, the thing is, if you think about how the technologies has evolved, and I mean, I know for some of you guys, this is the world you've kind of grown up in, but for like people like Chris and myself,
Yeah.
Where this all kind of started, like, there was like microarrays, and then we kind of then ultimately moved to bulk sequencing, and then now have moved forward. If you really think about this, if you're like kind of looking at a tissue, or let's say in this case, you have, a bunch of fruit together, and if you just kind of mishmash it all together, that, make a smoothie, that's kind of bulk RNA, right? And so you just kind of have an idea of what's going in the whole group. You don't really know necessarily where it's coming from. And then, obviously the technologies have evolved where we can actually kind of harness, various labeling techniques to actually delineate, oh, well, these RNAs or proteins are coming from a specific cell. And so that's obviously the single cell transcriptomics revolution. And now you can actually associate that even more clearly in a way to know where these are coming from in a specific area of space. And so these are kind of the spatial technologies. So you can see how much more information you can derive, and then in organs that are quite appearing on the surface homogeneous, but really are quite heterogeneous, where this can actually be quite groundbreaking. And so, obviously I'm a little biased, so I'm just showing you here some livers, but this is kind of the liver architecture. And, on the surface, well, it's like, oh yeah, this all looks the same. But I'm here to tell you that it's not, that you do have some of these structures, but actually all these hepatocytes in between are quite different from each other, and the endothelium are different, and the stromal cells are all zonated. So now how this spatial organization impacts not just normal homeostasis, but also affects disease response and injury and regenerative response, which is unique to the liver, is also kind of a black box right now. And so something that, these technologies really can change things. And if you really were to think about the general direction of things here, you can see how, we started off with the big revolution in the 19th century, which, I know sometimes, going back to, old texts or things like that might sound boring, but you have to think about what was happening in the basically 1860s, 1870s. All of these companies, chemical companies, were making all these new dyes actually to dye fabrics, to make different colors for clothing and for things like this. And people in the medical community were just grabbing this stuff to say, oh, well, what does this do? What does that do? And they were discovering, like, well, they stain tissues differently, and essentially led to this revolution in histopathology. Obviously, in the late 20th century, with the advent of antibodies that allowed you to target specific proteins, you can have immunohistochemistry. This has evolved into the late 19th and the late 20th and 21st century, really looking at molecular pathologies. This has now moved into digital pathology, and so we'll kind of go into that right now. And so, I mean, where this has really pushed the field forward is, I'm also a clinician. Besides running a lab, I work as a liver doctor, as a hepatologist. And usually the way we've thought about disease is, you have a patient with disease X, and everyone, you just kind of throw everyone in that bucket. So you just have one treatment for a patient with autoimmune hepatitis, and that's what it is. But the reality is that autoimmune hepatitis is probably very heterogeneous. We kind of knew that as clinicians, but we just didn't have the tools really to delineate that. And there's probably many different subtypes, probably respond to treatment differently. We'll have higher rates of risks of relapse, things along those lines. And, if we actually have proper biomarkers, proper diagnostics, we can actually delineate what bucket these patients belong into, and then better tailor those therapies for them. So instead of one treatment for all, you can actually have a personalized treatment for one, ideal for their situation. So, basically one of the key challenges right now in the field has been that in order to better understand what is going on in disease, you actually have to first have a robust reference of what is normal. And this is something that's often missed actually in the single cell space, the spatial space, is people really just jump into the disease states. But the reality is you really need to have a robust reference for normal. And the problem is that, first of all, when you get tissues, most normal people aren't signing up. It's like, oh yeah, take some liver tissue from me. Why would they volunteer for that? And so that really creates a real challenge, because most of the tissues that we even reference as saying it's normal is not really normal. So it's something to be very thoughtful about. The other thing is, and I'm sure you guys see this all the time in your studies, like you do a cancer study. And so what do? They do single cell RNA-seq or bulk. They'll do it from the tumor, and then they will do tissue adjacent. So I can just tell you that tissue adjacent tissue is profoundly abnormal, and it may be controlled genomically for that patient, but everything else is pretty abnormal. And so again, it's the best reference you might have in that context, but it's important to note it's not normal. So the other thing to point out is, where are you getting this normal tissue from? Since most live patients aren't volunteering to have their normal tissue evaluated, usually you're getting them, again, from these disease states, or you're getting them from autopsy programs. And, I don't have to tell you guys that someone who's dead for 2 days, the tissues might not be robust. And just, even in rapid autopsy programs, when you see that in a paper and things, a rapid autopsy means generally done within 48 hours. Really rapid is in 24. And it just, to give you an idea, organs like the liver are profoundly abnormal with basically any ischemic time. So it's just to put that in mind. And that's true for a lot of organs in the body. So again, just something to point out when you read these papers, and you kind of just gloss over that. And it's kind of an important thing to keep in mind. So I'll skip over this. So, basically, what tools can we actually leverage as a community to really understand what's going on at the cellular and subcellular level, and actually kind of dive into and not like avoid these questions as it relates to tissue and cellular homogeneity, heterogeneity. And really the spatial proteomics and transcriptomics technologies really give a window now to look at this. And you obviously have to always keep in mind what the advantages are and limitations are of each of the individual approaches or technologies that you're doing as they might apply to the different tissues that you're looking at, because each tissue is going to have its own caveats or things attached to it. So basically, this is kind of a figure kind of encompassing the variety of different multi-parameter techniques that are currently available in the marketplace. On the left is kind of the resolution in nanometers, and on the bottom is the number of markers or genes that are available to look at this. And you can see here that this is looking at kind of the metal antibody or metal-based approaches, which I'm going to talk about, both imaging mass cytometry and MIBI. Basically, there are fluorescence-based approaches, and they all essentially are using essentially like a fluorescent barcode type of approach to actually help delineate that. Now, as a result of that, they're actually able to get greater plex of markers. But again, there's certain caveats with those technologies as well, which the metal antibodies don't suffer from. I mean, I can just mention them here. Like, again, the liver is just to pick on it. The liver is incredibly autofluorescent, like incredibly autofluorescent. I don't know if you've ever looked at an animal, you just throw some UV on it, the liver literally just glows green, and it's intense. So pretty much anything less than 550 is useless to use as a fluorescent probe. And the techniques to eliminate that autofluorescence are somewhat limited. So again, it depends on your tissue. Brain also super autofluorescent. So, again, there's some vulnerability for these techniques if you're going to be using that in that context, which on a metal-based approach, you don't have that problem because there is no autofluorescence. And I'll go into that in a second. And then obviously with DNA barcodes, you can actually go, to a far greater plexing depending on the technique that you're using to kind of look at that barcode itself. So I'm just going to talk to you about two techniques that we collectively been working on. One's called CosMx, and the other one's molecular cartography. And I'm just going to briefly introduce a collaborative project that Chris and others are part of. I'll acknowledge that in a few slides. But again, as I mentioned, it's really important that we have a robust reference data set of what's normal human tissue. And basically, the goal has been to develop a data set from normal human adults, both men and women, with a variety of different ancestries. And you can see here that we're looking at a variety of different tissues from the human body. And within that, we're not just also looking, the goal is not to look, the small intestine is huge, the colon is large. So we're also looking within the different clinical and anatomic regions of those tissues as well, because those tissues aren't just homogeneous colon. There are different areas that clinical anatomic designations actually have histologic, molecular, cellular differences within them.
Can I ask, what is the average number of spatial samples that you have per person? Does it seem very extensive?
Yeah, I mean, the goal ultimately is that we're going to have, 12 patients, 6 women, 6 men. And then from within the individual patients, it depends on the tissue. So prostate, the whole prostate's been going on there pretty much. But for like colon, we just have a few segments from that specific area. The thing is, within a specific zone, the heterogeneity that we've been seeing in a particular anatomic area has been somewhat okay and not creating a significant statistical issues. But the thing is that, and we've been seeing that also between patients as well. So I think a lot of the heterogeneity we're going to see is as we move through this. And the question is, we've been very careful to pick areas within the very tissues that are very defined. So like from the colon, we've been doing cecum or, rectum or things like this, which are very designated things. Which we haven't run, like if we ran transverse colon, the transverse colon is large, probably about maybe 30 centimeters. I have to imagine if we picked one area within that area versus the other, we're going to get different results. Things that we also haven't explored is like in the liver, the liver has 5 lobes. The thing is that I have to imagine, although no one's ever shown this, so I'm just saying this, but if you start getting into the nitty-gritty, I have to imagine the lobes are not the same. That the lobes are somewhat different. Their blood flow is not exactly the same, and areas that feed into the liver, into those certain areas is also different. And so I have to imagine that there's difference between the lobes. We're not getting into that fine granularity. The other tissue we've kind of staying away from is like we're going to look at the brain. But like the brain, we spend this whole project in the brain because I mean there's all these different nuclei or regions which have inputs, outputs, and all of this, and we could probably spend 2-3 years just working on that alone. So yes, all great questions. Say this again? Oh, none of these are autopsies, so I should be very careful. This is all either biopsy or surgical material, but normal. We're not doing like, oh, we have colon cancer and then we're taking this area. These are patients that, for whatever reason, have had either resections or biopsies for other reasons that we as clinicians in the group would not believe would be an issue. And we've been doing this in very close collaboration with the pathology team and have brought in domain experts from each of the individual organs to ensure that, we have complete agreement on the clinical pathology side.
Can I ask, what metric do you use to determine heterogeneity, or like the extent of the heterogeneity?
When you say like, which mean cellular heterogeneity?
Yeah, I guess so.
Yeah, I mean, I think it's a fair point in the sense that, I mean, look, every cell is going to be a little different from each other. I mean, I think the thing is that if you took like a root mean of like the various expression levels within the cells, and they're extending far beyond that. So, I mean, if you look at, let's say, the liver, for example, in a lobule, which goes from like the central vein to the portal vein, there is somewhere around maybe 50 to 80 hepatocytes that extend in that. So if you were to try to delineate what's going on, the hepatocytes that are directly adjacent to the central vein, they are profoundly different, like profound from all the other ones. So that single cell would be its own group. If you keep going down though, they kind of, if you start doing this, unless you start going really fine, you'd see that there's hard demarcations at the protein and RNA level that break it up into 7 groups. But if you're going to ask me the computational method that we use, that would fall more into Chris's bucket than mine.
No, okay, I mean, that makes sense. I didn't know if it was like expression like heterogeneity or like actually like spatial like.
No, it's expression. It's based on expression of RNAs and proteins within those cells themselves. That's how we're defining.
Yeah.
Defining heterogeneity.
Okay.
And I mean, every once in a while you'll find a stray cell that it's like, what the hell are you doing there? Because you're kind of like an outlier. But that might also be, something related to the processing of the tissues or things like this. Cool. The other thing to point out is all the techniques that I showed you is really being done on FFPE. So, not perfect tissues. Some of these are in frozens, but most are not.
Thanks.
Okay. So this is just, this is an old slide, but it just kind of gives you an idea of all the different tissues and where we've gotten to this point. And, actually just to say, I should have updated this. Pancreas is already done. We're right now running a few additional runs for the liver, colon, bone marrow done. We're right now running lymph nodes and things like this. So we're already on that next, like lymph node, rectum, duodenum, or rectums twice, because evidently we have 2 rectums. No, it just should be 1. But anyway, okay, we've basically done the whole GI tract. And so I just want to acknowledge everyone who's been doing this. Obviously, Chris's lab and Drew and Park and his lab, Shauna, myself, and then the epigenomics core, and then our colleagues over at NanoString. And the one people I realized I did not put on here and I should have is the whole pathology group, which is absolutely been essential. So what can you do with this and what the benefit of this is? So basically, So this is an appendix. I'm not expecting you guys to know what an appendix looks like, but this is what your appendix does look like. And this appendix is normal. And you may ask, how do you get normal appendix? Well, actually, about a third of the appendixes that are removed in the context of acute appendicitis are normal. That's a good thing, because you wouldn't want them be erring on the other side and letting them explode in the patients, trying to avoid doing surgery. But if you start looking at these, you can see what the autofluorescence is. But this is, if you use a variety of morphology markers, this is what the parenchyma looks like here. And using this approach, you can actually ascertain the boundaries of the cells using a segmentation algorithm. Okay. And this, we could talk about offline, because there's several different segmentation algorithms that can be leveraged for this. But the reason why this is quite important is here, this is going to allow us to visualize. Now, we're looking at different RNAs. We have what the boundaries of the cells are. And so this tells us where these RNAs are located. And then it's really about now throwing this data back into the similar pipelines as what you do for single cell RNA-seq, because the thing is that what RNA and the number of counts of those RNAs are on each cell. And so you can use that to actually do clustering analysis. And then this allows us then to remap those different clusters back onto those slides. And so by doing that, you can see where these cells are located based on the clustering analysis that has been performed. And this is what the architecture of the appendix does look like. This is going to be stroma. These are the different crypts that exist here. There's going to be a bunch of immune cells here. So this all looks appropriate. And you can do this type of analysis across the whole GI tract. And if you just look at 3 different tissues here, you can kind of see what that looks like here for the ileum, colon, and appendix. And what you can do this can actually show you some real cell-cell interactions here. This can look at the protein level. See that this is looking at different receptors. You can then use the RNA to look at the different signaling. And the thing that's different here is if you really think about the interactions that when you do single cell RNA analysis, the ligand receptor interactions that you see, that's kind of all imputed, right? You don't really know if the cell, if you see signaling and you see this like, oh, there must be some ligand receptor interaction with this other cell, but you don't really know if those cells are actually in any spatial relationship with each other. Well, that's not true here anymore, because you could do that same analysis, do the predictive ligand receptor analysis, but you can then actually do the sanity check to see where this is in relation to each other and actually hone in on the different interactions that are actually real rather than the ones that are imputed. So the other thing that this type of analysis allows you to do is basically a niche types of analyses. So I'm just going to zoom in here, and you can see here. I know this is small, but if you can, these are the different cell types that are clustered here. This is within what appears to be a Peyer's patch within the small intestine here. And you can see that if you do an unbiased nearest neighbor type of analysis or neighborhood analysis, you can see you can actually generate these different niches that are here. And you actually can see how they're correlating with the different cell types here. But you can actually then look and decode what are the different cell types that are actually driving the kind of unbiased analysis that was provided here in the niche analysis. And you can see here that there's a variety of different B cell interactions, while actually in this outer area is much more of these NKT T cell that are living there. And again, this is something that happens to be known. So but this actually is showing this absolutely directly. Then you can start asking bigger questions. Before we were talking about cell-cell interactions, but you can actually look at how different niches or groups of cells actually might be interacting with each other, both within and outside of the niche themselves. And it kind of examine that based on the strength of the different arrangement of the cells in relation to each other within the niche and the niches around them. And So basically, this would give you essentially a prediction of the types of interactions that are being present from within the different cells. Not unsurprisingly, usually the most likely interactor of a cell X is going to be with itself or cells like itself. But then this can actually allow you to identify ones that are a little more surprising, or maybe not surprising, but like here you have macrophages, interacting most with arterial capillaries, which is something, again, that is known. So again, it's a good sanity check, but this gives us the hope that then you can actually identify novel interactions or novel organizational interactions that haven't been really discovered before. I'm going to skip over this part.
And just to ask, that interaction is based on like closest cell type to like.
So some of the analyses can be run that way. Some of them can actually be based on ligand, different ligand receptor interactions, but based on the spatial organization within that. It depends which algorithmic approach that you try to take.
Okay.
And obviously, the more you get into the weeds, the more I would say speculative those are, that one would probably want to test those. So, well, you're going to want to test these all anyway, but absolutely. The other thing that this also shows you that, if you actually look, one of the things we wanted to do is, because we're making a GI map of the different tissues, do you actually see that given that they're all derived from the same germ layer, same origin developmentally, are there certain interactions that are, conserved across the different tissues? And you can see that there are, and this is comparing the appendix to the ileum, which are actually quite different tissues. But what's interesting is you can then also ask questions like, if this is looking at the cecum and comparing it to colon cancer derived from the cecum. And what you can see is that there are, some interactions that are in common, but you can see here that there are some that are enriched in colorectal cancer that aren't seen in normal cecum. And some of these actually have been reported before, but some of these others have not. And so this kind of gives us now also some, obviously if someone wanted to pursue this, it opens the door to start dissecting this more to try to see if this is part of the regulation of these specific cancers. Let's get over this. So, I think the goal here is, or I should say, the goal here is to actually try to link the molecular and cellular changes that we've seen previously in, single cell approaches, and now try to connect them to the spatial organization that's present within those tissues. You actually can, obviously, if you get deep data from single cell RNA-seq, you can link this now to a spatial approach, which obviously does not have the same depth of targets as that. And you link these together to actually develop a really detailed map of the microenvironment in that tissue, in the case that we're looking at in normal tissues, but with the goal, obviously, to extend this to disease as we move forward. And so, basically, this is, kind of where we are currently, but, obviously what we're trying to push into the future. So I'm just going to give you guys quickly just two very quick vignettes, just to give you an idea of how we've been thinking about this and what we've been doing with this. And I will do it quickly, so I apologize. And the main purpose is just giving an idea of what we've been doing. So as I mentioned, I am obviously biased and interested in the liver, although I do work in a lot of other infectious diseases and other organs. And one disease that we work on is a disease called Alagille syndrome. So Alagille, no reason for you guys to know this, it's rare. But basically there are two common developmental defects in the liver as it relates to bile duct forming. One is that the external bile duct, the one that emanates out of the liver, just doesn't form. Or if it forms, it just is very small, too small for the patient, or it just disappears as they, in the first few months of life. And so that disease is called biliary atresia. Again, makes sense. No, atretic, or it means lack thereof, basically, bile duct. Now, there's another disease, that's the extrahepatic, the large bile ducts that emanate out of the liver. The other one is this disease, Alagille, so very small intrahepatic bile duct, or lack thereof. And so because they don't have a bile duct in the liver, they have problems, because when patients have biliary atresia, what they do is they do surgery on the patients, and they just slap the liver onto the intestine. So, okay, there might not be a tube to connect, but they just connect it directly, and that'll actually rescue about 30% of those patients, and they won't need a liver transplant. But these patients have no bile duct at all inside, so they kind of suffer, and really the only treatment here is a transplant. And the thing is that this is a genetic disorder. So about 95, 96 of these patients have a defect in jagged, and about 1 to 2% have a defect in the receptor for jagged notch 2. And maybe I should have put on here what that pathway looks like. I'm hoping you guys know, but jagged notch is like one of the canonical, pathways. And to make a long story short, this is an autosomal dominant disease. So if you're just missing one copy, you don't produce enough jagged, or you don't have enough of the receptor, you kind of don't have bile ducts inside your liver then. So basically, one of my colleagues, Stacy Huppert, she's at Cincinnati Children's. What she's done is she actually has made a mouse that has the same defect as these kids. And I hope you can see this here. You can see here, this is what the bile ducts look like if you label them. See how beautiful it looks. It just creates like a beautiful tree in the liver. And if you look here at P30, you kind of see it's like it's not really making it the whole way. And I know it's if you notice here, the ducts are very thin and very refined. Here, they're kind of like almost bulbous and quite big. And it's because like it's actually defects or cavities that are forming that are forming, and it's very thread-like. And so really it's not capable of getting rid of the bile for these kids. And so the mouse phenocopies this well. And it wound up identifying something pretty wild. Okay, so this was published now, 6 years ago. And what she found is if you make the defect only in the hepatocytes, okay, and this is ultimately going to impact the bile ducts and the hepatocytes, and knock out the ability to respond to notch or jagged, I should say. You can see here no bile ducts again, right? But look at 4 months, everything looks normal. So what the hell is happening in this mouse? And what she found is, as opposed to people, they're total knockouts. In the mouse, she can knock it out in one cell type or the other. And by doing this, what she actually found was that in animals where only a certain cell type it's knocked out, where the hepatocytes will become cholangiocytes and rescue the phenotype and will transdifferentiate. And so the question that she has had for the last decade, when she first identified this, is what is in the microenvironment that is allowing this to occur? Obviously, in people, this is a problem because they're complete knockouts. But if we actually had an explanation of this, maybe we can actually help treat or rescue these kids. And so basically, God, I'm going too long on this part. We identified a cell type in my labs basically now 4 years ago, where essentially there are fibroblasts that live around these bile ducts. And it basically turns out that this is what's creating the microenvironment, that niche that is required for telling the hepatocytes, hey, you should become bile duct cells to rescue the problem here. And so that's the key cell. And basically, we show that if we manipulate or target that specific cell type and prevent the stroma from actually doing its job in the niche, it actually prevents this rescue. Okay, so that's the summary of, I don't know, 10 years of work. So basically, what we try to do is leverage spatial transcriptomics to try to understand this process dynamically as it's occurring in real time, because we have data from single cell, but we don't know where these cells are in relation to each other. And the markers, the thing is, to identify the cell type, we need like about 100 different targets to actually identify this rare cell type. And I have to say, the technology that we're using here is only limited to 100 markers. So really was not able to kind of dig deep into this, but with the advent of the next technology, which I'll have like exactly probably 5 minutes to talk about, basically that technology will allow us to delineate the cell type. But using the single cell RNA-seq data, we were actually able to develop very specific, maybe 2 or 3 RNAs to at least delineate each cell type that we were interested in. But again, we don't have enough room for signaling or crosstalk or any of these things. And so the thing is, though, we were able to identify some things that was quite surprising and not something we expected. And it's not something maybe because people haven't looked that deeply, have seen before. So this is healthy liver. And as I told you, the liver is zonated. And remember, what do I mean by zonation? So this is just looking at two RNAs, LIS2 and CYP1A2. And you can see that they're expressed in different areas here of the liver and within the lobule. So one of these kind of surrounds basically the portal area, and the other one is mostly in the central venous. And you can kind of see it creates this pattern. The thing that we see in Alagilles, we actually see that the expression level is lower, and the areas are kind of collapsed within that itself. So it's telling us that the zonation or all of the composition of the liver and the way it's organized is a little different in Alagilles than it is in normal healthy liver. The other thing that we saw is, so SPP1 is a marker specific to cholangiocytes. And you can see cholangiocytes are the bile duct cells, and you can kind of see where those are in healthy liver. Now, the dogma in the Alagille field is really there's no bile ducts, because remember, they can't have them, but they actually are there. We can detect cells that are expressing these markers. It's just they're not doing anything. They're not able to properly do their job. And again, it's because they're not organized properly within the liver itself, because they should be organized in like this very, clear circular pattern. Here, they're just like all over the place. So basically, the other thing, and this is not surprising, we looked at jagged one. There's no antibody for that, but these techniques, because you're looking at RNAs, works beautifully. And you can see that there's a lot of jagged one in normal liver. And you can see here that the amount of RNA is much lower. Not surprising, they have a defect in our jagged one. They're all, they have a haploinsufficiency. And basically, again, I already showed you that the cholangiocyte geometry is completely messed up. Not unsurprisingly, there's more upregulation of cell cycle markers in the diseased liver, and it makes sense. The liver is essentially dying, and so the cells are trying to compensate for this lost mass. And we see that there's a huge expansion of that niche that I was describing earlier of these fibroblasts that we had discovered in this area. It just, again, because they have a defect, they can't help in this environment. They're there, they expand, they're responding to the injury, but because they can't have the proper, because they have a mutation, they can't respond in the appropriate manner. So the other thing, so we had shown that there's some alteration in TGF-beta expression, and SERPINF1s and tagline are hallmarks of downstream signaling of TGF-beta. So I know I told you I couldn't look conclusively, but we were able to look at a few. So I'll skip over this piece, but even with just 2 RNAs, we actually were able to delineate each of the individual cell types in the liver, which I know sounds kind of crazy, but because it was so clean and specific, and this technique, again, advantage of this technique is it has very good specificity. We were actually able to delineate these different cells as well as the different subtypes within it using a variety of these different markers. And if we just kind of map back, again, like similar to what we did before, this is what liver tissue looks like. And basically, this is what the disease tissue, and I know this is looking at very large groups, but we're actually able to delineate, 14 different cell types within the liver tissue, even using just a 100 RNA plex technique. So, imagine as we move to 1,000, 10,000 type RNAs, it's really going to allow a lot more depth. Now, obviously, this is in a very well-studied tissue that we've put a lot of effort into delineating the different populations using single nuclei RNA-seq. The thing that we found, though, is that the different interactions doing this is all the different cell types that we see here, are quite different between human liver and Alagille's liver. And so basically, I mean, I'm sure you can guys see this, but there's actually a profound alteration in both the interactions between both immune and epithelial cells here. And the reason why this is interesting to us is, at least in this disease, no one's ever actually looked at all at the immune compartment in this disease. And so we've actually gone back to the data sets that we have to try to examine this more. deeply to try to understand what the interaction is of the immune system within this. The last thing I just want to say on this, which this also helped us. So again, we have the human tissue, which is the real thing, and then we have the mouse. And one of the questions always in the field, which no one really has an answer to, is how well does your mouse model phenocopy the human disease? And the thing is that if you do some bulk RNA-seq, it's like, yeah, it looks messed up. Okay, it's the same. But if we actually now here are pulling the the expression of each of the individual cells, their arrangement, all of these different components, we were able to produce different maps. And actually, what we are actually able to find is actually it phenocopies disease quite well. And really, there's only one cell type here, which is one of the zonated hepatocytes, which actually seems to be quite different in the mouse as compared to the. And this is true both on its expression, based on its organization, and things like this. Now, what that might mean, we're trying to figure out. But I think this is also has hope for kind of helping us all in the basic science community for delineating what is or is not different from the model that you're using to study this disease. Because obviously, if you know that, then you can actually better understand what the limitations are. Okay, so okay, with 4.5 minutes left, I'm not going to just dive into all of this on COVID, but what I just wanted to show you guys is, another technique, and I don't know how familiar you guys are with this, and I just want to introduce it so you just can get familiar with this as well. So basically, this technique is called imaging mass cytometry, and the reason why this has been so valuable for us, as I mentioned, the liver is painfully autofluorescent, and so are a variety of other tissues that I unfortunately also have decided to study. And so this has really made fluorescent-based approaches quite painful. The other thing is that if you imagine you lose pretty much half your fluorescent spectrum because of autofluorescence, you really are limited in the number of antibodies you can use due to the issue known as spectral overlap. The thing is that even with multi-parameter spectral flow cytometry and imaging, you're still going to be limited to 5-7 antibodies. It's not like you can throw in 40 antibodies at a time. Now, what imaging mass cytometry does to get around this problem is you essentially just take a variety of different isotopes of metals that are not present at any concentration in the tissues that you're studying. So in nature, there is about a grand total of about 41 isotopes that you can obtain at high purity that are not actually present in tissue. Obviously, if you're going to look at iron, that would be a problem because we've got a crap ton of iron in us. So that's not going to be helpful. But if you do this for isotopes that aren't present in us, then that in the antibody that's tagged, that area, if you're looking at by mass spec, that area that you looked at has to be coming from the antibody that you labeled your tissue with. And so this has allowed us to go up to 41 different parameters. In this study, we just did 38 different parameters. But basically, this allows us to actually get very refined data. So one of the good or bad, the resolution's a micron. So it's subcellular-ish. But it overall is pretty good. And this is looking at just the global architecture, in this case of the lung. And so this allows us to ask questions about the organization of the vessels, the size, the alveolar area, which is the air spaces, how much collagen there is, and how intense or dense that collagen might be. And just to, giving you here on the right here, I should have shown more pictures, but this also kind of gives you an idea of, what these, what these images might look like. But essentially, just similar to what I described before, where the cells are, what the boundaries are, what proteins might be there. And this allows you to do cell clustering analysis similar to before. And you can go dive very globally into, bulk cell types, but you can actually start, depending on the markers you have, define this with great granularity. And then similar to what we described, this allows us to now look at basically, different cell-cell interactions. You can create these different butterfly maps to look at this, and you can actually then answer questions about how this would be different between normal or different disease states. So my whole point is that, obviously, there is a multitude of these different emerging spatial technologies. The first one I was talking about that we've been working on, NanoString, can do at the RNA. Basically, you can tag RNAs or proteins with this, and using that technique, you can get 1,000, 6,000 different RNA targets, protein level 64 right now, but it'll go into the hundreds ultimately. And essentially the computational approach, obviously might be a little different from technique to technique to technique in some of the limitations, but overall the way of thinking about it, once you have these cells segmented and moving on, the different imaging analysis techniques actually do some similarity. So with that, I can take any questions that you guys might have. The one thing I should just do here once I get there, and I wasn't going to present all this, I just was going to take a few things, was I just wanted to acknowledge this has been a lot of different people. I want to acknowledge the people within this lab and Olivier Elemental lab. Acknowledge the pathology team to contributed to that part. I'd gladly take any questions.
Thank you so much. And we'll also get these papers and the slides up on the website as well. And we're at the top of the hour. I know some people had to hop off. But any other questions? So Dr. Schwartz will be available. He's on campus. He's an expert not too far away.
Yep.
I actually had a question about the Alagille syndrome mouse model. Did they look at what ratio of cells needed to be mutated in order for you to see the rescue?
Right. So, I mean, the thing is that, when you're doing this with an albumin, when you're doing this with a CRE or CRE-ERT2, so the CRE, I mean, if we're targeting cell type X, in this case, the cholangiocyte population or compartment developmentally, that's going to be hitting the stem cell. So pretty much all of the cells derived from that population are going to be impacted because the recombination event is quite efficient. If you do this with a tamoxifen inducible CRE, like a CRE-ERT2, that is quite variable depending on which tissue you're targeting. In this case, we have recombination rates in the high 90s. So it just happens that it's very efficient. Now, there's another study, has nothing to do with this, where we're looking at the liver sinusoidal endothelial cell compartment and its impact that those endothelial cells have on liver regeneration, injury, things like this. Our CRE-ERT2, our inducible CRE, was at best 60% efficient. And the impact to that was that, you couldn't see a phenotype, even though we know if we hit 80 or 95%, it's very profound. So to answer your question is, as it references human patients, look, human patients is 100%, right? So, we're really trying to understand that disease context and not the threshold question that you're trying to ask. The one thing where it is relevant we're working on right now is from a therapeutic standpoint, what percentage we'd have to have. And the answer there is, we think we only need to repair or a small percentage, because in the liver, because it has regenerative capacity, it'll actually have a competitive advantage in the liver, and it will grow out over time if you can keep the patient alive and comfortable and happy during that period of time. So that's the rationale we're using right now.
That was actually my follow-up question.
Oh, okay.
Yeah, makes sense. So are you pursuing?
We're pursuing that. We got the NOAA on a grant, notice of award on a grant yesterday to do this from the NIH. So this is what we're hoping to do over the next 4 years. So 4 or 5 years. Yeah. Absolutely.
So my question is much more general, so I completely understand the answer is just complicated. But since you started with with generative model generation of liver, I've been wondering if since liver is self-regenerating, when a patient gives a healthy sample, would the same niches be regenerated as they were originally?
Okay. Yeah. So that question has not been studied in human really well. But in mouse. So this is maybe getting into a little nuance. If I remove 2 of the 5 lobes of the liver in the mouse, those 2 lobes are not regrowing back. What happens is the 3 remaining lobes will hypertrophy, get bigger, and there'll be proliferation of hepatocytes and other cell types, the endothelium and everything. And that's called hyperplasia. So that's what occurs. So the reason why I'm just being careful is like if in a salamander or axolotl, if you chop off its arm, an arm grows, but it actually, the organ that was lost regrows back. Humans, no organ in human has that capacity. But the one thing the liver is endowed is the cells do have the capability to do what you just said. That niches, everything will be restored. Hasn't been shown in human, but I suspect that it would occur because classic thing is we do what's called living donor transplant. I give 2/3 of my liver to someone who is very sick. That my liver, that third I've left, will grow back. Now, not the lobes that are gone, but the ones that are left will get bigger. And if you look by biopsy, it appears indistinguishable from normal liver tissue. But no one's looked rigorously with any of these techniques. I would imagine that there'll be subtle differences that we would be able to detect, but I can't say that for sure.
And usually it is like 2/3 of, like one side or, and not separate samples from like different places, right?
Say this again. So if I were to burn part of my liver, like I stick a probe into my liver and I burn 10% of it, the rest of the whole liver as a whole will expand to replace the mass that's lost.
Right, so what I'm asking about, if there is any merit in essentially building like an organoid, which would be started from like pieces of all of the lobes, as opposed to taking like 2 lobes and like.
Okay. So the answer is I'm not doing this, but there is a group at the University of Pittsburgh that actually is a company where they're doing this. Actually, they're taking about what the equivalent would be about like 0.5% of the liver mass, and they're actually putting it in lymph nodes, which seem to support as like almost like a bioreactor, and you get like a mini liver in there. And so they've done this in pig, and they've done this in actually 15 pigs. It was part of their preclinical. They're actually doing clinical trial right now. It's a phase 1/2 clinical study in humans, and we'll see if it works. But their data looks quite excellent. So yeah, quite excited. Yeah.
I don't know if there's an answer to this question, but how do you deal with the like trade-off between the really high resolution and the amount of area you can cover? Because I mean, I saw you had a ton of pictures or images, right? So like.
So each of the technology. I mean, I think you're asking a few different questions. So each of the technologies has its own different liabilities, advantages, and things like this. So like imaging mass cytometry or MIBI, these are techniques where you label and then you come in and you burn an area, and that's what you're assessing by mass spec. So there's 2 costs in a way, if you think about it. One of those costs is in the labeling of the tissue. The other one is in the acquisition time, which is at a fixed rate. So then you're really just balancing how heterogeneous you think the tissue is in order to get enough power to actually comment on what the differences are or how heterogeneous the tissue is so you can capture all the cells. And that's just a judgment call right now. I mean, in a more perfect world, we'd actually have some data so you can actually be like, oh, I need to acquire X amount of surface area of tissue. But because there's been so little data before, it's been hard to do that. Some of the technologies, though, you just label the whole slide, and the acquisition is, I don't want to use the word virtually free. And so that, you get a lot of cells and a lot of surface area in that. Now, I think you have a second question there, which is, there's also a computational cost here and time involved. And so how many cells, how much tissue do you need to acquire? And I don't think we have a good answer for that. I mean, all I can say is, and obviously Chris can jump in here as well, what we've seen is we need more. Like we have not run into a situation. It's like, well, we could do with less.
Yeah, more. I mean, I think a lot of there's been some papers that looked at this at bulk RNA-seq in early 2010s, 2011s, and there you can get to homogenization or saturation really of signal because you've homogenized the tissue. But that requires, I think, hundreds of millions of reads. And so in this case, we don't quite have that yet with spatial, but I think but also there's a lot more to see too. That's like a homogenization across a pretty simple transcriptome, but you're not looking at spatial information where you'd want more depth to be able to make sure you've mapped, say, ligand receptor interaction. So I think, yeah, more more is better.
Yeah, I just don't think we've gotten there yet. I mean, there will be a point at which we do, and I mean, particularly as these technologies, God willingly, get cheaper. I think there will be a point, it's like, well, we don't need to do that, and the computational costs will be the issue. But like right now, it's not the computational cost that's driving the costs here in these processes. So, I mean, I also think there's no data sets. It's also, sometimes you can build off data sets that are in the community. And so what you need to get in order to answer your question can be smaller, given what's already out there, but there's nothing out there. So you can't kind of leverage or, leverage that additional data to kind of shed some light on what you're doing.
That's really cool. I work with Jan from Seek.
Yeah.
Who's a neighbor's person, so
Yeah.
We're doing like diffuse large B-cell lymphoma, and I think we're doing spatial metabolomics, like IHC overlap during our peak.
What technology are you using for the spatial metabolomics?
We use DESI.
DESI, okay. Yeah.
So we can do same tissue.
Yep. Yeah.
IHC and metabolomics.
I mean, there was this crazy paper that maybe I should. So they did you name every omic technique, they did it for the liver. You name it, they ran. And I mean, it was quite interesting. I mean, the problem is, and this as well, is this paper was really trying to push DESI down to half a micron. And as you know, the smaller you get, the less the less specificity and sensitivity you have on maybe I should say sensitivity on the metabolites. So there is a give and take that you guys have. What resolution are you doing on DESI?
We're starting at 60 microns.
60 microns, yeah. So that's not. So I mean, this is the problem. I mean, if you think about this, so that is multicellular. You're doing a multicellular analysis. So in a tissue like the liver. Or frankly, if you're doing diffuse B-cell lymphoma, I mean, that's a big chunk of the lymph node that you're looking at. So, but I think it's great. I mean, it's obviously, there's still going to be, those lymph nodes are large enough so that you'll have some heterogeneity within that.
Yeah, a ton. And also, I mean, I wanted to ask, because I know you're doing lymph nodes or you showed your.
We are.
Or
yeah, it's actually, I don't know where it is in process. It was supposed to go on, if I'm remembering right.
No, it's really cool. I mean, I would love to look at it if you have any chance of making that available, but it's really.
Yeah, we'll talk offline.
Yeah. No, that sounds great. I really appreciate it.
Absolutely. Well, Chris, thanks very much. I'll stop sharing and
I'll put the recording. Let's go onto the YouTube channel for the course. Thanks so much for coming to speak. I look forward to seeing you soon. I'm sure at some venue, and I put links to the papers in the chat, and we'll put them online as well. And great. Thanks, everybody. And some of the homework's been emailed out, and we'll have people present more next week. And thanks, Dr. Schwartz. Thanks.